Very good evening, children. Are you able to see me? Can you hear me? Can you all hear me? Are you able to see me? Vashini is there, Kadiravan, Balakrishnan, Kishore, Balamurugan, Deepak is there, Mahadev Krishna. Why only Kadiravan is responding today? What about others? Are you all there? Is this topic transport in plants, is that okay for you? Is this topic transport in plants is okay for you today? You want to do chemical coordination. Why, Vashini? Any specific reason for that? Vashini, any specific reason for that? Because morning itself I have asked you to give suggestion for the topic. Nobody has responded to that. Any specific reason is there? We'll go with that, no problem. Any topic it is fine. Okay, so what I will do today, yes, welcome Chari. Okay, no issues. Kishore want organ and organ system in animals. What about for Kadiravan, any topic is fine. Okay, so what I will do, anyway we'll start the topic transportation in plants. I want to concentrate more in physiology. That's why I've taken plant physiology. In that already we have completed photosynthesis process. The next topic is about we'll complete this and then I'll do anyway we have to do the control and coordination, that also I'll do it. So now I'll do with transport in plants. Okay, so today's class I want to give some assignment to you all. I want to give some assignment. What is the assignment? All of you take one A4 sheet with you. One A4 sheet with you, or else if it is a plain paper, if you have one side paper, that is fine. Now what are all I am going to discuss today, all those key points, So this topic, it contains about 67 slides. I want all the key points to be noted in a single sheet. You got it? All the points, summary points, not all the points what I am telling, the key summary points. I want you to do it in a small piece of paper, and then I want you to revise that lesson with the help of that particular paper. Are you all clear? So all of you take an A4 sheet, and all the key points what we are going to discuss, all those points you have to note it in the one side itself. Don't go for second sheet or third sheet. Only one sheet for one lesson. That is for summarizing. Why I am telling that? Suppose if you want to study the topic, you just go through that one page. That is more than enough. No need to go through the textbook again and again. Are you clear, children? That is the reason why I want all of you to write the summary points. So we'll start the topic. So dear children, today's topic we are going to talk about is transport in plants. Transport in plants. Like we have studied about the circulatory system. In the circulatory system, the materials are transported within our body with the help of blood and lymph. The transportation takes place within our body with the help of the blood vessels and the heart. Similarly, the materials required by the plants are also transported within the plants with the help of xylem, phloem, veins, veinlets, with the help of the root, stem, with the help of leaf. A lot of materials are transported within the plants. Now let me define what are the materials that are transported in plants. The materials that are transported in plants, it includes water, minerals, nutrients, plant growth regulators. 4 things. These are the important material that are transported within the plant. First, let me tell you from where to where the water is transported. The water, it is present in the soil. Water is present in the soil. Because if there is a rainfall, the water percolates down into the soil. So water, it is almost the water, it is present only in the soil. There are certain plants which absorbs water from the atmosphere. Example, the vanda, the epiphytic plants. It absorbs water from the soil, whereas all other plants, it absorbs water only from the soil. From the soil, the water is absorbed by the root hairs. Absorbed by the root hairs. Root hairs are the only organ that absorbs the water. In the lower class, we might have studied that water is absorbed by the root. But exactly the roots are absorbed by root hairs. I will explain you what are root hairs, the root hair structures, all those things in the next class. From the soil, the water is absorbed by the root hair. From the root hair, the water is transported to the root xylem. You got it, children? The root hair absorbs the water from the soil. From that, it transports that water to the root xylem. From the root xylem, the water molecules, it will move in upward direction. That is, it enters into the stem xylem. So with the help of xylem, the water is transported throughout the body, throughout the plant. This is how the water is transported. From the soil, it enters into the root hair, root hairs to the root xylem to the stem xylem. Through the stem xylem, it reaches the whole plant. Similarly, the minerals are required by the plants for various process, for various physiological process. It requires the minerals. So from where these minerals are present, these minerals are also present in the soil. The rocks contain minerals. Due to weathering process, the rocks, it will become, due to weathering, it will become soil particles. All the minerals that is present in the soil, except the few it is present in the atmosphere, like nitrogen molecules. Most of the new minerals are present in the soil in the form of salt. Already what is there in the soil? Water is there. So mineral salt are there. So what will happen if you add salt? We add salt into the water, it gets dissolved. So always when plant absorbs water from the soil, it also absorbs the minerals along with it. So minerals are transported throughout the plant with the help of xylem. The same way how plant absorbs the water, the similar way all the materials are also transported within the plant. Next is about the nutrients. We know that by a process called photosynthesis, the plant produces nutrients. Where exactly it produces the nutrients? In the leaf, there are special tissues are there. Those tissues are called as mesophyll tissues. Those tissues are called as mesophyll tissues. Those mesophyll tissues, it does the process called photosynthesis. During photosynthesis, plant produces carbohydrate. The plant produces the carbohydrate. That is glucose, which will be converted into starch. So that starch molecule from the leaf, it will be transported throughout the plant. So that transportation, or in turn we use the word translocation. The translocation of the food particles, it is done with the help of phloem vessels. With the help of phloem. Similarly, the plant growth regulators. The plant growth regulators are the substances which helps in growth and development of the plant. They are also called as phytohormones. Some of the examples: auxins, gibberellins, cytokinins, indolacetic acid, ethylene, abscisic acid. These are examples of plant growth regulators. These plant growth regulators are also transported within the plant with the help of xylem and phloem. So these are the materials that are transported in the plants. What are the materials? Water, minerals, nutrients, and plant growth regulators. Next we are going to talk about the direction of transport. I have told you the materials are transported within the plant, but in which direction they are transporting? There are certain materials which are transported in unidirectional transport. Some of them it is multidirectional transport. So what is this unidirectional transport? The term uni means single, right? So you can see it here. The soil contains water and minerals. So the root hairs, it absorbs the water and minerals. From the root hairs, the water and minerals, it enters into the cortex of the cell. From the cortex to the endodermis. From the endodermis, it enters into pericycle. From there, it enters into the xylem. From the xylem, the root xylem, the water molecules, it move in upward direction. So here you can see that the water molecules always moves in upward direction. All the water molecules and minerals, it will move from soil to the upward direction to all the parts of the plant. So that is called as unidirectional movement. From the soil, the water and minerals are absorbed and the water is transported to the aerial parts of the plant. That is called as unidirectional transport. Next is we have the multidirectional transport. In the multidirectional transport, the food and plant hormones, because the plant hormones are produced in certain regions of the plant. From there, it has to reach all the parts. Similarly, the sugar molecules, the glucose, the sugar or starch molecules are produced in the leaves. But you need to understand that all the leaves will not do the process called photosynthesis. There are some aged leaves, it will be there in the plant. Those aged leaves, they will not do the photosynthesis process. Similarly, the young leaf, the foliage leaf, it will not do the photosynthesis process. Only the photosynthesis process are carried out by a matured leaf. So from that matured leaf, the food material, it has to be transported to the young leaf, which will be on the top. Root requires the food material, the stem requires the food material. So from the leaf, the food molecules are translocated to all the parts of the plant in various directions. That's why it is called as multi-directional transport. The sugar molecule, the glucose molecule, a starch molecule that is produced from the leaf, it will be transported to the flower, transported to the bud, transport to the young leaf, transport to the aged leaf, transport to the stem, transport to the root. So the food molecules are translocated to all the parts of the plant. That's why it is called the multi-directional transport. Next we are going to talk about what are the means of transport. What are the means of transport? So far children, do you have any doubt? Till now, do you have any doubt? Children, do you have any doubt? Vashini, your doubt is where is plant growth regulators are formed? There are specific regions are there in the plant. For example, there is a plant hormone which is called auxin. That auxin, it is produced in the tip of the leaf. Tip of the leaf. From there, it is transported throughout the plant. So like that, each and every plant growth regulator, it has certain specific regions are there. From those regions, it will be transported throughout the plant. Are you all clear? Okay, next doubt, Kajiravan. What is the difference between the root xylem and stem xylem? There is not much difference. Even I can say there is no difference at all between the root xylem and stem xylem. It is just a continuation of that. Since the xylem is present in the root, it is called root xylem. The same xylem, it is present in the stem, it is called stem xylem. There is no difference there. Shall we move on and learn about the means of transport? Means of transport. Are you all clear with that? Balakrishna, do you have any doubt? No doubt. Ashini, no doubt. Kajiravan, okay. Okay. Deepak, do you have any doubt, Deepak? Deepak Ishwar? Okay. So we move on to the means of transport. Means of transport. What are the various means of transport? Diffusion, osmosis, facilitated diffusion, active transport, passive transport. These are all some of the means of transport. Just like that, the material will not be transported from one region to another region. Whether it is food material or it is water or minerals. Just like that, it will not get transported. There are certain forces that are involved in transportation of materials. Now let us learn about what are the means of transport. Diffusion. We have learned that the definition for diffusion in the lower class. That is the movement of molecules from the region of higher concentration to the region of lower concentration. Mainly solute molecules. The movement of molecules from the region of higher concentration to the region of lower concentration until it will become equilibrium. Until it becomes equilibrium. That is called as diffusion process. Again I will repeat, diffusion is a process by which the solute molecules are transported from the region of higher concentration to the region of lower concentration. Even diffusion occurs in gases. So the movement of molecules from higher to lower concentration, that is called as diffusion. and there are certain medium or characteristics are there in the diffusion process. Temperature play an important role in diffusion. That means if the temperature is higher temperature, the diffusion it will be faster. Lower temperature, diffusion it will be lower. It will be slow. Surface area. If the surface area is large or the region of absorption is more, the diffusion it will be faster. That is the reason why the small intestine you have lot of inner foldings, right? That is called this villi, which increases more surface area. When larger surface area is there, more diffusion it will be there. Diffusion it will be faster. Similarly, the concentration gradient. The concentration gradient, if it is higher, diffusion it will be faster. The size of the particles, smaller the size, faster the diffusion. The size is smaller, diffusion it will be faster. Similarly, the medium of diffusion is also very, very important. If the medium is a solid medium, diffusion it will be the slowest, very, very slow. If the medium is a liquid medium, it will be faster. The same, if the medium it is going to be the gas medium, it will be the fastest. These are all the characteristics of diffusion. Diffusion means, you know it, higher to lower concentration. For example, the water that is absorbed by the root air, from the root air it enters into the cortex. That movement it occurs by diffusion process, because the adjacent cell it has higher concentration of water, whereas the next cell it has the lower concentration of water. So from there it will be easily transported. So higher to lower concentration. That is called as diffusion. Next is facilitated diffusions. What is facilitation? Facilitation means someone help. That is called facilitation. Here the molecules are transported. The molecules are transported across the membrane. Across the membrane with the help of certain protein channels. They are called as carrier proteins. If you see the membranes, the biomembranes of the plants and animals, there is the cell membrane or the membrane that covers the organelles. All those membranes, in their outer membrane they have the protein channels like this. They are called as carrier proteins. With the help of these protein channels, they can easily move inside and outside. So here you can see that this it will come and attaches here, and with the help of that it will move inside. So this is called as facilitated diffusion. Here minimal expenditure of ATP it is used here. The energy is used for transportation. So the transportation of material with the help of a protein channel, that is called facilitated diffusion. Next is porins. What are porins? Porins are huge pores that are present in outer membranes of plastids, mitochondria and certain bacteria. This large pores it allow the molecules up to the size of small proteins. So these are the pores, they are called as porins. With the help of porins, the materials or the molecules are transported within the membrane or within the cell or even outside the cell. That is about the porins. Next we are going to talk about the aquaporins. What are aquaporins? Aqua means water. There are integrated membrane protein channels which mainly transports the water and small solutes across the membrane. These are all specific pores. They are called as aquaporin. This aquaporin it will transport only the water molecules and small solutes. This type of aquaporins are present in the case of bacteria, plants and animals. Next we are going to talk about the types of transport that occurs in the plants. There are three types of transport occurs in the plants. One is called uniport. Uniport. Here only one type of molecule, it moves inside or outside the cell membrane. Either a particular group of molecule, it enters inside or it goes outside the cell. That is called unipotar. Uni means single. Unipotar. The next is called symport. Symport. In the symport type, two molecules move inside. Two different types of molecules. Two types of molecule together, it cross the membrane in the same direction. Same direction. Can you see it here? Same direction. Two different molecules, it moves inside. That is called symport. Antiport. Here two molecules, two different molecules, but they are moving in outside opposite direction. One it goes outside, the other one it enters inside. That is called as antiport. Uniport means single molecule. Symport means two molecules at the same time, same direction. Antiport, two different molecules moving in opposite direction. This is about the types of transport. Next we are going to talk about the active transport. Usually in the diffusion process, I have explained you the movement takes place from higher concentration to the lower concentration. Higher to lower concentration. When something has to be moved from higher to lower concentration, energy is not required. Suppose we want to drop something, you are on the second floor, you want to drop something from there, energy is not required. Whereas if you want to lift something from ground floor to the top floor, you need lot of energy, because you have to move against. Similarly, the active transport, it is a mechanism which molecules move from lower concentration to higher concentration, lower concentration to the higher concentration. When something has to move from lower concentration to the higher concentration, that is called against the concentration gradient. What is concentration gradient? Higher to lower. When something is moved against the concentration gradient, that is from lower to higher, that is called active transport. For active transport, here you can see that these are all the higher concentration molecules, whereas outside it is lower concentration. So the lower concentration, from the lower concentration to higher concentration, molecules are moving inside. They are moving inside with the help of energy. Here high expenditure of energy is required, ATP is required. When something has to be transported against the concentration gradient, more amount of ATP is required. That is called active transport mechanism. Active transport mechanism. This is the comparison of different transport process. Here you can see that requires specific membrane proteins. The membrane channel is what we have studied, porins, aquaporins, carrier proteins, like that. Highly selective, transport saturates, saturate means stops or not, and uphill transport, that is against concentration gradient, that is called uphill transport, and requires energy, ATP, yes or no. So this is the comparison of different transport process so far what we have studied. Okay children, now make a note of this. I'll give you 5 minutes time. We'll go for a short break. Just copy this slide. This is a very important slide, and after that I'm going to explain the next process, osmosis, and types of osmosis, and what will happen when cell is placed in different mediums, and potato osmoscope experiment, thistle funnel experiment, plasmolysis, imbibition. These are all other forces are there, and after that I'm going to explain to you the plant water relationship. Okay children, now just copy this slide. I'll give you 5 minutes time. After that I'll come and explain you about the various other process in detail. Okay children, if you have doubt, you can post your doubts now. Children, let us continue the topic. The next topic is about osmosis. What is osmosis? It is same like diffusion process, where molecules it will move from region of higher to lower concentration, but it is through a semi-permeable or differentially permeable membrane. There is no difference between differentially permeable and semi-permeable, both are same only. What is semi-permeable membrane? The membrane that allows only certain molecules to move across. That is called a semi-permeable membrane. For example, if you take a cell, the cell has the outer membrane. So this outer membrane, cell membrane, it is a semi-permeable membrane. That means it will allow only certain molecules to move inside. Only the molecules that are required by the cell, that alone it will be transported inside or outside the cell. Similarly, the vacuole is the structure that stores the cell secretions. Those vacuoles are surrounded by a layer called tonoplast. Even that tonoplast, it is semi-permeable in nature. It will not allow all the molecules to move inside. So here, the movement of molecules, if it takes place from higher to lower concentration through the semi-permeable membrane, that is called as osmosis. When the water molecules moves from higher chemical potential to the lower chemical potential, that is also called as osmosis. When this osmosis it will stop, here you can see that the molecules are moving from higher to lower concentration. When it becomes equilibrium, then it will stop. So always molecules will move from region of higher to lower concentration until it will become the equilibrium. Till equilibrium or till it becomes equal potential, it will keep on moving. Next we are going to talk about the types of osmosis. Based on the direction of movement of water in a system, in a osmotic system, there are two types of osmosis is there. One is called endoosmosis, the other one is called exoosmosis. The term endo means inside, exo means outer. Endoosmosis, here the osmotic entry of solvent in a cell or a system that is placed in a pure water or hypotonic solution. Again I will repeat that. Here the molecule move into the cell when it is placed in a pure water or hypotonic solution. That is called endoosmosis process. In the endoosmosis process, what will happen? The water molecules, when you keep a substance in a water molecule, those water molecules, it enters into the cell. That is called endoosmosis. It moves inside. For example, when you place a dry resins, in a water, usually some of them, they have the habit of eating the dry grapes, even the badam, they'll put in the water. So what will happen? The water molecules, when they put the dry grapes inside the water molecule, what will happen? After some time, it imbibes the water and swells up. That process is called, the swelling up process is called turgid. That process is again an endoosmosis process. Next is exoosmosis. In the endoosmosis, the water molecules enters into the cell, whereas in the exoosmosis, the water molecules it withdraw from the cell. When it will withdraw, when you place them in the hypertonic solution, when a cell is placed in hypertonic solution, it loses water. When you place the cell in a hypotonic medium, hypotonic solution, it gains water. The exoosmosis, when water is lost from the cell or a system, it leads to a condition called plasmolysis. What is plasmolysis? Here when you keep the cell in a hypertonic solution, what will happen? It loses the water. When water is lost, what will happen to the protoplasm of the cell? Here, the thing which is present inside the cell wall, this is the cell wall. Inside that, what is present? The cell membrane. Inside the cell membrane, what is present? Here you can see that the cell organelles, cytoplasm, nucleus. When a cell loses water, it will become shrink. Similarly, when you place a cell in hypertonic condition, what will happen? It loses water. Due to loss of water, the protoplasm, it loses the water, it will become shrink. When it shrinks, what will happen? It will move away from the protoplasm. It move away from the cell wall. All the thing what we are seeing it here, this is the cytoplasm as well as nucleus, everything loses water and it will shrink. When it shrink, what will happen? Usually the protoplasm is attaches to the cell wall. When it shrinks, what will happen? It leaves the cell wall. Attaches to the cell wall. When the water loss, it leaves the cell wall. It gets separated from the cell wall. That process, it is called as flaccid. That condition, shrinking, is called flaccid, and that process, it is called plasmolysis. Plasmolysis means it loses water, it shrink, and it move away, it pulled away from the cell wall. That process is called plasmolysis takes place. The same cell, when it is placed in hypertonic solution, hypotonic solution, what will happen? It gains water. So this is called plasmolysis. It loses water, it becomes shrink. When the same cell, the plasmolyzed cell, if it is kept in a hypotonic solution, more water enters inside, the cell regains its structure, it will become turgid. Turgid means swells up, and it comes back into the original shape and size. That process is called as deplasmolysis. When a resins, when a dry grapes loses its water, that condition is called plasmolysis. When you place that in water, it gains water, right? That is called deplasmolysis. That is about osmosis. Now let us see about the types of solution based on concentration. There are three types of solutions are there. One is called hypertonic solution, hypotonic solution, isotonic solution. What is this hypertonic solution? In hypertonic solution, there will be the condition, the hyper means high, tonic means solute. Here a strong solution, that is a low solvent or high solute or low water potential. A strong solution, it attracts solvent from other solutions. So high solute, hyper solute, this is hyper solution condition. Can you see it here? Outside the cell it will be higher, inside it will be low, the solute concentration. So it attracts solvents from other solutions. When it attracts, it loses water. Similarly, hypotonic solution, low conditions. Here inside a weak solution is there. When a solution is weak, it diffuses water out. That is the reason why when you place a cell in hypotonic solution, it gains more water. So inside the solute concentration is higher, outside it is low. That is called hypotonic solution. Isotonic, iso means similar, identical solute. Two solutions having the same concentration. So there will be no movement. The net movement of molecules, it will be zero. There is no movement of molecule. That's why it is called isotonic solution. Okay children, have you understood the three types of solution? The hypertonic solution, the solute concentration is very, very strong. So it attracts the molecules from other solution. That is the reason why inside the cell, the water molecules are attracted by the hypertonic solution outside. So it takes more water. Whereas in the hypotonic solution, the solid concentration inside the cell is higher, so it attracts the water molecules from outside. That's why the cell gains water and become turgid. When the condition is both the solute concentrations are same inside the cell and outside the cell, then what will happen? No movement, the net movement of molecules will not take place. The condition, it will be zero. No movement of molecules takes place. These are all the types of solution based on concentration. Next we are going to talk about the potato osmoscope experiment. Potato osmoscope experiment. Okay children, till now do you have any doubt? Till now do you have any doubt? I will clarify your doubts. If you have any doubts, I will clarify your doubts. Have you all understood the concept? No doubts for Rashini. What about others? Adiravan? Yes. Charvi? Okay ma, very good, very good, very good. So now I'll just explain the potato osmoscope experiment, and after that I'm going to explain about the thistle funnel experiment. Okay children, now just copy this diagram. What will happen if we keep cell in hypertonic solution, isotonic solution, hypotonic solution? Okay children, just draw this diagram. Sometimes in your board exam, a school exam, they'll ask you to draw a cell in isotonic solution, hypotonic solution, like that. So I'll give you 5 minutes time. I want you to draw this diagram, and after 5 minutes I'll come and explain you about the osmosis, potato osmoscope experiment. Okay children? Children, I hope you have drawn the diagram. The next experiment what I am going to talk today, it is about potato osmoscope experiment. Here I will just explain you how the process takes place. How the process takes place. Okay. So here for this experiment we have to take a large size potato, and peel off the skin. Just take out the outer skin, and then at the center of the potato, make a hollow cavity. Like this you have to make a cavity. It should be at the center. It's not like, should not be cut like this. Should not be cut like this. Center is here, it's not cut like this. Center, it has to be a hollow cavity, and then take a large size beaker containing colored water or water, and inside the hollow cavity of the potato you have to, up to certain level, you have to put the sugar solution, concentrated sugar solution up to certain level, and mark the level with the help of a pin, a small needle. Now just cut the base of the potato and make it flat so that it can easily fit into the bottom of the beaker. Take the beaker should contain some water. If it is a colored water, it will be fine. Now place that potato inside the beaker containing water. So the water level is up to this, right? You can see that water level is here and the potato is here. The potato is flat, inside hollow cavity is there, and inside the hollow cavity what is there? Sugar solution is there. Just leave the setup for about 6 hours, and after 6 hours you can see that from the initial level, this is the initial level where you have kept the first pin, right? There will be rise in the level of sugar solution. The concentrated sugar solution, it become dilute sugar solution, and then mark that with another pin. That is the final value. So what this indicates, the water that is present here, slowly it enters into the center hollow cavity, and it converted the concentrated sugar solution into dilute sugar solution. Here the movement of molecules takes place, but not all the molecules enters inside. The reason is, here this process is not diffusion. Even though outside the potato the concentration is higher, all the water molecules will not enter into the hollow cavity, because the potato cells of the potato, it act as a semi-permeable membrane. It allowed only the certain molecules to enter inside. That is the reason why there is a slight rise in the level of sugar solution. This is the experiment to prove that osmosis takes place in living cells. This experiment is called potato osmoscope experiment. The next experiment is called thistle funnel experiment. Thistle funnel experiment. So what is the thistle funnel experiment? Here we have to take a thistle funnel, this is a thistle funnel, and invert the funnel. Invert the funnel, and here at the bottom there should be a membrane. This membrane, it should be taken from the animal skin, that membrane. Inside the funnel, sucrose solution, it will be there. Sucrose solution, it will be there. Got it, children? A thistle funnel, the bottom of the funnel, it will be covered by a membrane, an animal skin, and inside sugar solution is there. Here we have to place that thistle funnel in a beaker containing water. Beaker containing water. And here on the top, this water molecules which is present inside here, it will try to enter inside the sugar solution. So the water molecules try to enter inside, that is called osmotic potential of the water. Any molecule, if it has to be transported from one to another region or one place to another place, it should have the potential. That potential is called as osmotic potential. Here the water, it has the osmotic potential. So the water try to enters inside the thistle funnel. But similarly you can see it here, there is also a pressure here. That pressure, it is called osmotic pressure. So at one point of time, from the bottom the osmotic potential it will be there, and from the above osmotic pressure is also there. So this side osmotic pressure, here osmotic potential. So what will happen? There will be no movement in the molecules, because osmotic pressure and osmotic potential, these two, it will not allow the water molecules to move inside. The same concentrated sugar solution is maintained there. Even the osmotic potential is there, the osmotic pressure should allow the water molecules to move inside. If only the osmotic pressure is there, what will happen? All the sugar solution across the living membrane, slowly what will happen? It enters into the water. But here what is there? Osmotic potential is also there. Always the osmotic potential and osmotic pressure, it will work antagonistic to each other, opposite to each other. This is what the thistle funnel experiment explains to us. The next experiment, next is we are going to talk about the plasmolysis process. I have already explained you what is plasmolysis process. When you keep a cell in a hypertonic solution, it loses water and become plasmolysed. That process, it becomes flaccid. That process is called plasmolysed. So when you keep an isotonic solution, what will happen? The external solution balance the osmotic pressure of the cytoplasm. So it will be like more or less, it will be same. There is no net movement. Whereas when you keep a cell in a hypotonic solution, the cell gains water and it swells up. That process is called turgid. Turgid. When the cell loses water, it will become plasmolysed. Because completely it loses the water, it will become plasmolysed. This is called plasmolysis process. Next is the another force that is called imbibition force. I have given the example of the dry grapes, right? Similarly, the dry grapes as well as the imbibition of the seed, imbibition of the dry wood or dead wood is an example for the process called imbibition. So what will happen when you put a seed inside the water? Suppose most of the children, they used to have the habit of eating the badam. Badam, it is very difficult. It's very difficult to peel off the outer seed coat. So what the children used to do, they used to put in the water. They'll take a bowl like this, they'll put it inside the water. When you put that inside the water, what will happen? Slowly the water enters inside and you can see that the seed bulges. Similarly, when you put a seed inside the soil, during germination process also it takes up the water through the micropyle and it becomes bulge. Similarly, when you put a dry wood in water, after some days you can see that the dry wood, it swells up. What is the reason? The reason is imbibition process. Imbibition. What is imbibition? The water molecules enters inside the wood. The water molecules enters inside the seed. But that water, it is not dissolved. The water is absorbed by the solids, but it is not dissolved there. That results in swelling up. the water enters inside, உள்ள வருது water, ஆனா water என்ன ஆகலன்னா யூஸ் பண்ண முடியல, so it becomes swells up. That is called imbibition process. The colloidal system such as gum, starch, protein, cellulose, agar, gelatin, when you place it in water, it absorbs large volume of water and it swells up. It swells up. Any substance that absorbs the water, it is called imbibance, and the phenomenon is known as the imbibition process. Here also you can see that when you put the dry grapes inside the soil, inside the water, it takes up water, so the dry grapes are called imbibance. The process is called imbibition process. So these are the forces that are involved in that. Similarly, the imbibition, it is used in germination of seeds. When you put the seed inside the soil, it takes up the water. When it takes up the water, then only it swells up. So imbibition process, due to imbibition process, the water enters through the micropyle. There will be a small opening in the seed, that is called micropyle. Through the micropyle, the water enters inside and it bulges. This process continues. So what will happen? The seed coat burst. Whereas you can see it here. Can you see it here? This region, there is a burst in the seed coat. This is because this seed absorbs more water. Other seeds, it swells up. The swelling up mainly because of imbibition process. Similarly, the imbibition process, it also helps in absorption of water by the root hairs. The first process where water is absorbed by the root hair, that process is called imbibition process. Imbibition process. Next we are going to talk about the plant water relations. Plant water relations. First let me explain about what are the uses of water, and then I am going to explain about water potential, and then solid potential, pressure potential, and then I am going to explain to you about absorption of water. Okay children, so now what are the plant water relations? How water is used by the plants? So where and all water is used by the plants? Plants during photosynthesis, plant absorbs water from the soil. So where for photosynthesis what is required? Water is required. For growth and development of the plant, water is required. For transportation of materials in plants, water is required. The cell protoplasm, the plant cell protoplasm, 90% cell protoplasm, the cytoplasm, it contains water. That is the reason why always the plant cells are fleshy in nature, mainly because of it contains water. And also water is the one which helps in absorption of minerals from the soil. These are all the main uses of water. Almost all physiological process in the living organism, mainly in the plants, it depends on water. Transpiration, transportation, photosynthesis, growth and development, reproduction. For all these processes, seed germination, for all these processes what is required? Water is required. And also 90% of the plant cell, it contains water. Even the dead cells, the dry seeds, the woody parts of the plant also contains little bit of water in it. Some amount of water it will be there. And plant tissues need that water pressure or osmotic potential or water potential, it has to be there in order for the plants to hold this support. For rigidity, for to maintain the structure, to maintain the cell shape, water is required. That is the reason why when cell loses water, it becomes flaccid, it becomes deplasmalized, it becomes plasmalized. So water is very important to maintain the rigidity in the case of plant cells. And the plant cell wall as well as the vacuole, it holds water and it pushes outward, and also this water molecules, it keeps the structure of the plant from falling or wilting. If water is not there, what will happen? It will become dry up and then it will fall off. Even the plant cell wall and vacuole holds lot of water in it. With the help of water only, it can able to withstand the pressure that is from inside the protoplasm. Because always towards the cell wall there will be a pressure from the cytoplasm, that is also maintained by the water. With the help of water, the cell wall, the cell membrane, the vacuoles, the tonoplast, the outer layer of the vacuole, it has contains water which help them to prevent from damaging. These are all the main functions of water in the plants. Now let us learn about the water potential. What is water potential? The potential with which the water molecules move inside the cell or inside the plant, that is called water potential. This concept of water potential was first introduced by Slayer and Taylor in the year 1960. So every water molecule that is present inside the plant cell, it has certain potential to move. That potential by which the water molecules move, it is called as the water potential. And the potential, the water potential, it will be always expressed in pressure. That is called pascals. The unit is called pascals. The concentration of water increases the water potential. How? When more water is there, the water potential increases. The concentration of water increases the water potential there. Always a pure water, the osmotic potential is 0. There is no pressure. When pure water at standard temperature, not under any pressure, the pure water, the water potential, it is 0. Here, if a solute is dissolved in pure water, you can see it here, it is 0. When a solute is dissolved in the pure water, this whole water is pure water. So this pure water, the water potential, this is a symbol of water potential, is always 0. When solute is added to that, the concentration of the water decreases, reducing its water potential. Here, all solutions having a water potential than the pure water. All solutions have lower water potential than the pure water. So when something is added to the pure water, the potential, it is changes. Here, next we are going to talk about, these are all water molecules. The water molecules enters inside the cell. How it enters inside the cell? Through the aquaporins. Do you remember the aquaporins, which is present in the walls of the cell membrane? And through that the water molecules enters inside the plant cell. That process is called facilitated diffusion. When water molecules enters inside, it causes, so water molecules enters inside. Assume that this is the cell, water molecules are entering inside. So what will happen? The water molecules keep on entering inside. It exerts a pressure on the cell wall. When water molecules enters inside, the cell becomes turgid. What is turgid? Vingarad. Swells up. That is called turgid. When more water enters inside, it exerts a pressure on the cell wall. Thereby, it increases the pressure potential. The pressure potential increases. When the pressure potential increases, when water molecules keep on entering inside the cell exerts, the protoplasm exerts the pressure on the cell wall. That is called pressure potential. The water potential can be determined by two things. One is solute concentration or solute potential or pressure potential. So the water potential, relationship between the water potential, it can be determined, water potential is always determined, it will be like the solid concentration or solid potential plus pressure potential. So the water potential, which one determines that water potential? One is the solute potential and the pressure potential. Always the pure water, the water potential is 0. Why it is 0? There is no disturbance in the osmotic potential or pressure potential, there is no pressure in the solute potential. If a solute potential and pressure potential determines the water potential. So water potential is equal to solute potential and pressure potential. This is the formulae for water potential. So make a note of it. Now I am going to explain you about the solute potential and then pressure potential. What is this solute potential is all about? It is also known as osmotic potential. What is osmotic potential? The potential with which the molecules move. The solute potential, it denotes the effect of dissolved solute on water potential. So water has the potential. When solute is added to that determines that is called as solute potential. This dissolved solute potential, it have the impact on the water potential. Water potential is always, pure water is 0. When solute is added to that, the solute get dissolved in that water and it has the effect on the water potential. The pure water, as I mentioned there, the pure water, the water potential is always 0 to negative. When there is no solute, the solid potential is always negative. The pure water solid potential is always negative. So here the standard atmospheric pressure, the water potential is always equal to solute potential. That is the reason why the water potential is 0 in the case of pure water. Always the solute potential will be negative because the standard atmospheric pressure is also there. From that atmospheric pressure is also there, water potential is also there. So it will get always equal. You might have studied Thistle funnel experiment. The osmotic potential is from the down, upper pressure potential. So it will be always 0, negative. That is called solute potential. Next is called the pressure potential. So pressure potential, it is a force that work against the effect of solute potential. Solute potential is there, some forces it act against the solute potential, that is called as pressure potential. The pressure potential increases the water potential. That is the reason why when water enters inside the cell becomes turgid. That means the osmotic pressure is increased. The pressure potential is increased in the water. Water potential is also there for the water. Along with that, what is increased? The pressure potential is also increased. So it becomes turgid. So it exerts a pressure. That pressure is called as turgor pressure. The turgor pressure is mainly because of water potential along with pressure potential. When the pressure is against the cell wall, there is a positive hydrostatic pressure within the cell. That hydrostatic pressure, it is called as the turgor pressure. When the hydrostatic pressure is released, so water molecules when water molecules move out, the pressure potential decreases, so it becomes flaccid. When the cell swells up, you should assume that two things are increased. The water potential is increased, pressure potential is also increased. That's why it swells up, it becomes turgid. When cell loses water potential is losing the water potential as well as pressure potential. Because of that, it becomes flaccid. This is about solid potential and pressure potential. Okay, children. And now I'll just clear your doubts, and after that, if you have some time, let us discuss about the other topics. Okay. What is the difference between imbibition and endosmosis? Endoosmosis, that means movement of molecules within the cell. Always endoosmosis. Osmosis occurs in living cells. Osmosis occurs in living cells. So what will happen in living cells? A living cell means always we know that the outer membrane is semi-permeable membrane. The semi-permeable membrane, it allows only the molecules that are required by the cell. So in endosmosis process, the plant takes up whatever it is absorbed by the outer membrane. That is the cell wall or plasma membrane. Whereas imbibition process, it occurs even in dead cells. For example, the wood, the dry wood, it is a dead cell, but still if you put that in water, it swells up. So that is the difference between the endosmosis. Imbibition occurs in any cells, the dead or living cells, whereas endosmosis, it occurs only in living cells. And Kajiravan, you are asked about the agar. Agar, it is actually an algae, a substance extracted from algae, and that agar, it is used as a culture medium. Agar is a very good culture medium for growth of plant tissues. Agar is a culture medium which is from the algae. Madhav Krishna, your doubt is deplasmolysis and endosmosis are similar. The endosmosis it is a process by which the plant takes up certain substances. The deplasmolysis, it occurs after plasmolysis. You got it? After plasmolysis. What is plasmolyzed? The cell loses this water, it loses all energy. The water potential is not there, the pressure potential is not there. After that it gains that. That is called deplasmolysis. The process by which the molecules enters inside, that is called endosmosis. If something moves inside the cell, maybe the endosmosis process helps in deplasmolysis process. That is it is not similar. The endosmosis, it occurs in any cell, whereas the deplasmolysis process occurs only in plasmolyzed cells. You got it, Madhav? Okay. So another 10 minutes there, children. If you have any doubt, you can ask the doubt. And in next class, I will explain you about absorption of water, the pathway, theories of ascent of sap, transpiration, and closing and opening of stomata. There are a lot of theories are there. Factors affecting transpiration process, anti-transpirance, guttation process, direction of translocation. That's an important theories there. Munch-Marsh flows theory, source to sink. These are the topics what we are going to talk in the next class. Okay, children. If you have any doubt now, it is open for discussion, and I hope all of you have understood the concept, children. Okay. So now, if you have a doubt, I will discuss your doubt. Otherwise, just draw this diagram, because anyway I will ask you to draw this diagram in the next class. Just do the diagram, the same, all these diagram and also this flow chart. Draw it, and if you have doubt, I will clarify your doubt. Otherwise, after drawing, we will wind up the class. Okay, children. Do you have doubt? No doubt. Vashini has no doubt. Kishore Balamurugan has no doubt. And last class, do you remember I gave some assignment? Do you all remember that? There are few students who have not submitted the assignment. I hope the assignment is very useful to you. Am I right, children? Today morning also I have received certain assignment. I have got assignments from Kishore has sent it today, and Suparna Lakshmi already she has sent it. And Charvi have not sent the assignment. Am I right, ma? And Vashini, you have sent it. Yes, ma. Just copy this diagram. Those who have not submitted the assignment, please do it and send it to me. I will give more assignments like this. I want you to understand and write, not just copy it from the book, just understand what is there. That is very, very important. I want all of you to get the first mark in your class, in your school. I want that. Are you clear, children? Just draw the diagram. If you have doubt, I will clarify your doubt. Otherwise, we will meet you next class. Next class, I will inform you when we are going to have the next class. Okay, children? Thank you very much. If you have doubt, I will clarify it. Otherwise, thank you very much.